two people who are in the wrong meeting. I put a message in there to say that they were in the wrong one.
I've sent them like join requests.
Yeah,
Okay.
I'm here from the other one. I think the other guy's AFK.
Okay,
Cool. Just wanted to make sure everyone was in the right place.
we're recording anyway, so
So yeah, just to start off, are you able to explain to me, I guess, the math, the stats behind like the main test? Because I was trying to figure out all the stuff they were doing with let me see if I can find the section, but it was at the beginning of like their diagnostic session where they're like, hey, this is what we did. And I was like, well, why did they do that? I have no idea why they're doing that.
okay. The stats in here are probably a bit over your head unless you've taken a couple of stats classes. And that's okay. You should still be able to understand the paper and their conclusions and everything without that. But with that said, what they were trying to do, what they were measuring was the proportion of infections of this chytrid pathogen. So that would be their response variable. And then they measured a bunch of different possible predictors, right? So they had, what was it? Temperature, season, growth stage. So whether it was like larval or an adult, right? The year, latitude, longitude, and phylogenetic relatedness. So they measured all this stuff. And what might be getting confusing for you was that not all of those things wound up in their final statistical model. Does that maybe adequately describe your dilemma?
Yeah, I was kind of more specifically interested as well in. I give me a second to find it here because I just pulled it back up. Um, the phylogenetics part, they did something where the chi, not the chi-square distribution, PGLS. What's going on with that?
Okay, so that's phylogenetic generalized least squares regression. I would not get too hung up on it. What's happening there is they built a phylogeny and they're basically looking at the sum of the branch lengths between any two species and using that as kind of a distance measure for how related things are. Does that make sense?
Okay.
So it's a way to quantify phylogenetic relatedness in relation to other things in a phylogeny. So the smaller the number between any two species, the more closely related they are. And so it's kind of a measure. Imagine a phylogeny, and you just went with a ruler and measured the length between the most recent common ancestor of any two points, and that kind of shows you if it's properly calibrated to time, like they did here. It actually gives you a pretty decent estimate of how long ago those two species diverged from a common ancestor.
Got it. And they made their own phylogenetic tree, it looked like, right?
Yes. Based on, they basically used like a previously published tree. They then went in and like calibrated it to a molecular clock so they could kind of quantify how long ago these species diverged from each other.
Okay, thanks.
Yeah, so that's why the stats were so weird in here, is because they were using phylogenetic relatedness as a predictor, and it's not straightforward. It's not like in our R class, weight and horsepower influencing miles per gallon. It's a more complicated distance matrix they're using as a predictor. All right, so
I have a question about just about like how they like collected the data. So it just said that like they went to an area and they did an exhaustive search and then they swabbed, but like what did they just find, like the amphibians and then swab those, like all the ones they could find, or what exactly were they looking for swabbing?
So they did swab amphibians, as many as they could find at each spot. And then those little swabs got subjected to quantitative PCR. So they had primers that would detect the BD pathogen, and you do PCR in there and it tells you how much of the pathogen is present or if it is there or not. It's actually the same test they're using for like testing COVID patients, right? You've got special primers for COVID virus, you do a swab, and you see if it amplifies. So same sort of deal.
Okay, that makes sense. Mm-hmm. Thank you.
And their response there was just whether it was present or not. They didn't, use some sort of measure of how much of it was present on each amphibian, just whether it was there on them or not.
I have a theoretical question. I guess it's kind of related to the paper. If you wanted to test the amount of zoospores in the water, could you run a water sample and do PCR on the zoospores, the amount of zoospores in the water, or would they not? I guess would they survive long enough for you to be able to do that?
I think you'd run into a couple issues. qPCR is a good way of determining the absolute number of starting molecules of interest, not necessarily cells. So a zoospore might have multiple copies of that genetic region. And so if you say, oh, I found 6 copies of this DNA in this water sample, that may have come from 1 spore, it may have come from 6. And so it's kind of difficult. You have to jump through some serious hoops to be able to then go in and say how many cells that equates to.
Gotcha. Thank you.
There are ways around it. One, you can get a good understanding of how many copies of that particular stretch of DNA you're sampling for are found in each cell. If you're just looking at one species of chytrid and you take 1,000 of them and count up the number of copies by doing genome sequencing, you can get a good guesstimate, like, oh, they all have 2 copies, right? If you're looking for multiple copy number, TS region can vary from species to species. A lot of gene duplication events have happened since speciation, that kind of thing. So it can get kind of tricky. Another way you could do like flow cytometry, where you take your water sample and basically train a machine to recognize a zoospore and just run it through there, and it kind of catalogs every time it sees one of those. So per milliliter, and then you can kind of extrapolate out from that. Or there's fluorescent methods, so you could tag, that still actually gets you to the copy number versus cell conundrum. But you're onto something in that aquatic environmental DNA is definitely a very rapidly growing field, and they're using it to detect rare species. Even fish surveys, they're showing that you can get a better understanding of what fish are present just by taking a water sample and sequencing the DNA than you can by going in electrofishing and everything, because it's just not as efficient.
Yeah, I tried to write up a protocol to be able to do this in Brazil, and I was kind of in a field house in the middle of the rainforest and just didn't have the equipment, and I didn't know what I would necessarily. And equipment doesn't do very well in humid, rainy areas. So I was just kind of curious.
Yeah, I feel you there. I did some work in Costa Rica and Panama, and my laptop just died. It had like mushrooms growing out of it. The jungle is really hard on equipment, and keeping a constant power supply and everything like that for a -80 freezer can be really challenging. One thing I have seen people do is take water samples from remote field stations like that, and then they mix it into basically a buffer that kind of freezes all the DNA to where it is at room temperature, and then mail those back out to a facility to process. And so it is a possibility. I can upload a protocol that I use if you're interested for aquatic DNA sampling.
Yeah, that would be really interesting. Thank you.
Yeah. Any other questions about the methods here? A big portion, like table 2, this is kind of confusing, but they're showing all the different models that they tested. So all the different theories of how things like temperature and seasonality and precipitation might affect what's going on with this BD. And they're showing all of those models and then some criteria about how well those models explained reality. Okay. So don't get too hung up on it. That's just them kind of going into the weeds on how they picked their final model. And what was the final model that they found best explained BD prevalence? This is kind of the main result of this paper.
It says all evaluated models included phylogenetic relatedness among the species. So essentially, the chytrid affected each species kind of differently, and then the ones that were more susceptible were more closely related, was it?
Yeah.
And then there were some that, based on the tree, that just were not susceptible almost at all, it looked like. That, let me see if I can find it, the graph there. I believe they were towards the middle of the tree that they showed. Yeah, the ones towards the middle of the tree, like your Eupsophis vertebralis in that area, they showed like nothing. But then towards the top of the tree, there was a pretty big concentration of susceptibility to the fungus.
All right, so their final model, what they found, basically they're asking what determines where and which amphibians are getting sick from this. And they noticed a few things. One, phylogenetic. There was a strong phylogenetic signal. So certain species were more vulnerable than others, no matter where they were found. Okay. And then there were a couple other things that they found were really important for determining the spread and prevalence of BD. What were they?
Hey, Dr. Zong?
Yeah.
I'm thinking when I was reading it that it was that it had to do with, I remember there's a part where it's talking about where humans had been, like where humans were related, because not only they had been deforesting, but they had been making such a drastic change to their environment that caused there to be more BD, I think.
Yeah, and the way they measured a bunch of human variables, right? And they found that one explained it best. You remember what that was?
Was it climate control? Like the difference in the climate? How my takeaway from that, if it was the climate, is this just people polluting the air and having a drastic change on the weather or something like that? Is that kind of what you were looking for, or is it something else?
It's a way of measuring all of that with a single value that they found was very interestingly explained almost all of the human associated stuff. GDP, gross domestic product. You remember that from the paper?
Yeah, it
Yes.
was.
Yeah, I remember that.
Yeah,
with
and
the
so
with higher GDP.
they found they could measure these like 15 different human variables, or they could just measure GDP. So the economic development of the region was a strong predictor of BD prevalence. And then there was one more thing in their final model.
Was it variability in rainfall?
Yeah,
It seemed like.
the variability in precipitation. So you've got phylogeny, you've got, like their standard stuff. If you look at figure 2, the caption, model with the best fit, standard plus precipitation variability plus GDP, or times GDP. That was the model that explained everything best. Okay, and the standard was species, growth stage, season, year, and location. All right, so what they found was just by looking at phylogeny of these species and their location, that didn't quite explain where BD was being found. But when they added in precipitation variability and gross domestic product, suddenly their model's really, really strong. So this is a really cool incident of basically people just taking observations of where BD is found on what amphibians, measuring a bunch of stuff and then narrowing it down to what's actually important. Okay, so they're basically saying that we can predict the spread of this outbreak if we know the species, the precipitation variability, and the gross domestic product of a region.
So then, knowing like the information that we have now, is there like any kind of solution in sight that we know how to like stop it from spreading?
Well, model, we can change the species that are present. We could stop human development, or we could control the weather and precipitation.
So not really.
No. so I'm gonna say probably not.
Didn't it talk about how it was hard for them to determine some stuff because they could tell that it was also moving south?
Yeah.
How can they, I guess, measure that just by finding populations that haven't been affected yet, I guess? And
yeah, what they're saying is that the latitudinal gradient that they noticed, right? So location was important also. That was correlated with a potential ongoing spread from one of those regions, human-caused spread, so like people just moving it around. And so it was tricky for them to tell whether the signal they saw in latitude was really due to latitude or whether it was confounded with this potential like invasion. So I don't know that there's a way around that. This gets into the, to really test that, we would need 2 Earths, one to experiment on and one as a control. So they just make a nice note of that, saying like, okay, here's our model. It explains things really well, but take a grain of salt when you look at the latitude gradient, because that might be confounded with an ongoing spread of BD.
Geez, the things we could do if we had a control Earth to experiment on.
I would like to live on the control Earth and not worry about the experimental one. I just want to move there.
Same.
Well, okay, other thoughts about this paper? What would you take away from it, aside from getting depressed about all the amphibians going extinct?
Was I correct in understanding that at one point in the paper they were trying to basically assign a single value to each region that was basically calculating in every single piece of data they had? It was like phylogeny relatedness and the rainfall variability and the GDP and the latitude and longitude. They really just stuck that all into the same equation and put a single value in that, and that's how they correlated things?
In a way, not a single value, though. It's actually a matrix of all those values. And so behind the scenes in their model testing is some matrix algebra going on. And so rather than it being a single value, it's a conglomeration of all the values, and they're using that matrix as a predictor.
That makes more sense. I don't know how I didn't see that in the paper.
Well,
there was one point where it had like an
they
entire
just-
formula line.
Yeah, they just called it standard rather than, showing the whole matrix. And so in their formula, right before the results, there's like a formula in the middle of the page on the left column. That BD as a function of species, stage, season, year, lat, long, that was their standard. And so their final model that they found was standard plus the precipitation variability plus GDP. So yeah, the stats in this paper are definitely beyond like stat 2040 level stuff, if you've taken that. But it's the same sort of deal. They're doing a basic regression analysis. Just one of their predictors was actually a bunch of predictors in a matrix. Other thoughts, questions? What's the take-home message that you got from this particular paper?
The amphibians are dying.
Yeah.
But some species are more resistant than others.
Uh-huh. If you were a land manager working for like Department of Conservation in Chile, how would you use this paper?
You would probably be focusing conservation efforts into the areas that do have that major combination of the GDP and the variability in rainfall.
Mm-hmm.
Using that predictor, I'm not sure exactly what measures can be taken, but those that could be done that would be more resource limited would probably be best served by focusing on those areas that you can predict.
Yeah, for me, that's the take home also. As this is in essence kind of an applied paper, right? There's a real problem happening. They're looking for what explains where the problem is happening. They found some really good predictors, and so now resources can be focused on, say, certain species above others. or in certain regions, that kind of thing. So we have limited resources for conservation, especially for this particular subgroup, right? Just amphibians dying from this global disease. So if we're going to spend resources trying to prevent this, here's the best bang for your buck, kind of, is what they're saying.
There was a point in the paper where, if I understand correctly, it was saying that one of the major factors in a species being susceptible to the virus was how aquatic they were.
Mm-hmm.
Was there any other factors in the phylogeny that was deterministic on that factor?
See, I remember that. I don't remember anything else other than just phylogeny and the degree of aquaticness, I guess, did have a strong phylogenetic signal. So maybe that's really what's explaining it. It's just how aquatic they are. Well, it's reflected in their phylogeny. Trying to find.
I don't know if that was already mentioned, but they also said that the amphibians that are more resistant could be carriers of the fungus and introduce it to the ones that are less resistant. So if you're a land manager, keeping the ones that are resistant away from, if you can, the ones that aren't.
Yeah, so maybe that is like an achievable goal. We do it here in Utah, right? You take your boat out of the lake into another state. What's the first thing you got to do when you cross over?
Scald the crap out of it.
Yeah, right. You got to get it inspected. You got to like make sure it's nice and dry because we don't want to transport invasive zebra mussels around. And so, as annoying that might be as a boat owner, it is your duty as a good human to prevent the wholesale destruction of every lake you visit. And so maybe something like that, just working on preventing the spread of these amphibians from one region to another, could be a nice way of chipping away at this problem. I don't know what else you could really do. It's not like you can say no economic development can happen in these regions. We definitely don't have control of precipitation patterns. Maybe it is kind of a little bit depressing.
Talking about any method of environmental control that relies upon humans not being selfish is a very depressing topic of late.
Yeah, it's just not going to happen. That's the sad reality. It doesn't take many people not caring for it to become a problem. I mean, look at Utah Lake, right? Some people released koi into it like 40 years ago. It's basically the only fish that you can find. These disgusting carp that have taken over all the native fish. So it just takes one introduction here and there to really wipe out a local population.
This is completely unrelated to a lot of the other things being discussed right now, but that mention of the introduction of the koi, do we see a reduction over time in the introduction of new invasive species, or is it going up?
It's going up.
Well, crap.
Yeah, it's going up in pretty much every measure that you can take of it. For one thing, the climate is changing fairly rapidly to where species are able to move on their own a bit easier up and down latitudes. And then with increased just human travel, people carrying things from one spot to another, invasions are definitely on the rise all over the world. All right, any last thoughts about this paper before I move on to endophytes? All right, I am moving on.
So, are we gonna have a reading quiz this week? I mean,
Yeah.
we had it. Okay. For
Yeah.
tomorrow or for Thursday?
For Thursday, yeah.
Okay.
We'll have a reading quiz on the next paper, Christian et al. 2017. And today's endophyte lesson isn't very long, so we'll have time to kind of discuss where we're going. Kept it short since we're a bit behind. Okay, so we're talking about endophytes. Now we're moving into the section of mycology where we talk about symbioses. Okay, so we'll look at endophytes and mycorrhizae and lichens, that kind of thing. We spent a bit of time talking about fungi as the enemy. So to wrap that up, kind of shifting gears now, talking about fungi as friends. All right. What is an endophyte?
So endo would mean inside. So it's a fungus that lives in its host.
In a
Doesn't
plant.
it live outside of like a plant?
It's inside of a plant.
Inside of a plant.
Yeah. Phyte for plant, endo for inside. Now, rust fungus lives inside of a plant, but we don't call it an endophyte because it causes disease. Okay. Endophytes, as they're defined, live inside of plants, but do not cause any disease. So pictured here, where's my cursor? There we go. These are plant cells, and the squiggly dark blue lines, that's fungal hyphae. All right, so they're living in between all the plant cells, on top of, kind of surrounding, but they're in the plant tissue. Okay. So we're going to just take a brief look at them, see what they're doing. Pretty much that's about it, how they help plants, if not hurt them. Okay. So you may have heard of this drug in the top left, Taxol. Right, this is the most commonly used chemotherapy drug on the market right now. And it's really effective at stopping tumors. It was discovered in the bark of the Pacific yew tree. Okay. And so it's naturally produced. And so everyone went out and started cutting down a bunch of old Pacific yews to basically cure cancer. Okay. Maybe 15 years later, it got, we're looking for better ways rather than clear cutting forests of Pacific yew. How can we get this thing growing in the lab, that kind of stuff? And so they actually isolated where it's being produced, and it wasn't the yew tree at all that was producing this anti-cancer chemical. It was Taxomyces right here. So this is an endophyte that was actually living in the tree tissues, and that's what was producing this like wonder drug. Okay. So it was a fungus that makes Taxol. And so now we don't have to cut down trees anymore because we can get this fungus to grow in other species and extract the Taxol from it that way. Okay. As a matter of fact, we've even more recently been able to take the genes from Taxomyces, stick them into fungi that are easier to grow, like in a petri dish, and get them to produce Taxol. So endophytes are pretty important. A lot of people pay attention to them. They make a lot of neat compounds, and we've notice that every leaf we've ever looked at in the wild, I mean 100%, have endophytes in them. Okay, so they're in every plant you've ever seen. It's full of endophytes. So endophytes, fungi or bacteria, but we're talking about the fungi here. Fungi living inside of plant tissues, not causing disease. What's the point of them? What are the plants getting out of it? What's the fungus getting out of it? Any guess?
Just a quick
Fungus is getting nutrients, right?
Fungus might be getting nutrients, at least not very many. If it was getting a bunch of nutrients, we'd call it a parasite, because that would be damaging the plant.
And I assume it's just like how we have fungus living on our skin, that it would be providing some sort of protection against something worse.
Okay, so kind of like the commensal thing. Is that what you're talking about?
Yeah.
Okay. Other thoughts? What's the plant getting from it? What's the fungus getting from it? Why does every plant have endophytes?
Are these the variety of symbiotes that are fixing nitrogen?
No.
Didn't think so. Thank you.
Fungi can't actually do that. So I think it was Hayden talking about the potential commensal / mutualism. Can you elaborate on that?
I mean, I just kind of had a vague idea of how we also experience fungi. I mean, if I would think it would be similar also to like the relationships that like ants have with trees, in that the trees will allow the ants to be like taking leaves from it if the ants are protecting the tree, that kind of a situation.
Mm.
That
Yeah,
that maybe the endophytes that are living within the plant tissues are preventing other kind of disease.
Okay, so the endophytes, they're definitely not hurting the plants, or we would call them parasites. They're just hanging out, and in the process, maybe they're helping the plant in some way by maybe occupying a niche that a parasite would take.
Yeah.
In the Taxol example here, they're making anti-tumor compounds. Why are they doing that? Is it because they really care about human health?
That's a great question. I don't know.
The answer is no. Fungi do not care about human health. They're not aware of humans. But this anti-cancer compound acts as a deterrent for other parasites. It just happens to also be good at stopping tumors. So they're making secondary compounds that might be helping the plant, like prevent herbivory or prevent pathogens from showing up. Maybe they're just taking up that niche to help pathogens show up. What's the fungus getting out of it if it's really just kind of hanging out and helping the plant and not parasitizing the plant?
Perpetuation, a habitat to live in. It's what life wants, is to continue living.
But why inside of a leaf? What if I told you that almost all of these endophytes, if you were to categorize them based on their like major trophic group, it would be saprotrophs. What do saprotrophs eat?
plant
So are they
sugars
eating, like,
flowing
dead-
through the plant?
Detritus.
Dead stuff, yeah. So they break down dead stuff. They're dissolving cellulose, getting carbon from it, all that good stuff. So most endophytes are specializing in breaking down dead plant material. And yet here they are living inside of leaves, just kind of hanging out. So one hypothesis for what the fungi are getting out of it is called the latent decomposer hypothesis. What might that mean?
Vague idea. Dibs on this plant when it dies?
Dibs on this plant when it dies. That's exactly it. So if you're already in a leaf and it dies and falls off onto the ground, boom, you're already there. You're the first to the feeding frenzy. So that's the hypothesis here, is that what these things are really getting out of it, because they're most of the time they're just kind of biding their time, laying low, not really growing very much. They don't have like extensive hyphal networks inside of these plants. They're just kind of waiting around. The leaves die and they're already inside, ready to start digesting that. So, excuse me. How would you test this hypothesis? Last time I taught mycology. I actually had a group do this as their semester project, was test this hypothesis.
I actually have a quick question.
Mhm.
If they're not getting any nutrients from the plant, how are they surviving inside of it?
They're taking some like excess sugars and stuff, kind of same as the commensal fungi on your skin. It's just kind of like waste products and stuff, just enough to keep them in a dormant sort of state, maybe making some secondary compounds, but not actively really growing. So they're getting some nutrients, but it's definitely not enough to harm the plant.
Would it be possible to measure enzymatic activity of these saprotrophs on living versus dead tissue?
Okay, yeah. That's a bit more complicated than UVU students last year did. They basically went in and identified endophytes by surface sterilizing leaves with like bleach and alcohol, and then seeing what grows out of them, identifying those fungi, and then also looking underneath those same trees as the leaves started to drop and seeing if it was the same fungi. Okay, so they just did a correlation analysis, basically looking at, are the fungi that are in the leaves when they're healthy and happy the same ones decomposing them? Okay. And what you find, this is actually from a meta-analysis here, is on the left, these are common endophyte taxa, and on the right, common. And there's a lot of overlap. It's not 100%, but the same sorts of things that are abundant as endophytes do tend to be relatively abundant as leaf litter. So currently, from what I can tell from the literature, our best guesstimate of what the fungi are actually getting out of this relationship, the data seem to support the idea that what they're getting out of it is first to the feeding frenzy, right? They've kind of got dibs on these leaves. Okay, so they're hanging out there, biding their time, waiting for the leaf to die. Now, how do plants get endophytes?
I got a question about just before you go on. Sorry.
Yeah.
So are these types of endophytes, are they kind of territorial? So if another fungus comes in, do they kind of fight with it, or are they? Sorry, what was that?
They do. They have chemical warfare.
Okay.
Yeah.
So is that one way that you could test it? Is if you put another fungus on it to see if it would react with the plant?
Yeah, during my postdoc, I actually tried that because I had that same idea, looking at territoriality, basically. And the experiment didn't work. I kind of failed at it. But the idea was you take a bunch of endophytes out of leaves from the same species of plant and grow them in agar, and then basically put them on the same dish and see which ones outcompete each other. And then I was trying to see if it was the same ones when I stuck them on the leaf, but I don't know. I wasn't smart enough to pull it off. But that kind of test, I think, would be pretty useful, looking at how these communities form.
Interesting. It was just a question I had. Thanks.
Yeah. But that actually is a decent segue into how do these communities form? How do endophytes get into plants? What determines, right? You're a plant, you're growing. Which endophytes do you have? Where do you get them from?
I think that it could have come from the soil that they were grown in, because if the endophytes are decomposing that plant and then something new is going to grow out of the leftovers, the endophytes would still be there.
Yeah. Okay, so they could get them from the soil. Where do you get your skin microbiome as a human?
Can't they get them from the air too?
They could get them from the air.
Should be like, precipitation.
Yeah,
just like rain.
So rainfall, snow.
How species specific are these endophytes?
Yes and no. Some of them seem to be, some of them are pretty wide-ranging.
I would expect that the ones that are more species specific probably are straight up in the seeds of the plants when they're falling off the tree.
Okay. So we're talking about two different kinds of transmission, vertical and horizontal. Vertical transmission meaning coming from your parents. So the endophytes just packaged in your seed. You've got it from birth. Okay. Horizontal is you're picking it up from the environment. So after your seed lands, you get basically whatever endophytes are around. So that's two very valid ways of getting a microbiome, right? Humans are what, vertical or horizontal as far as our microbiomes go?
I want to say both.
It's horizontal for us. We don't have bacteria packaged in with our sperm or egg, right? Hopefully not, right? Probably wouldn't survive. Okay, so we're picking it up. We get our skin microbiome during birth through the birth canal, right? That's our initial seeding of our skin microbiome. We get our gut microbiome through milk and through just putting crap in our mouths because we're stupid babies. Babies like eat cockroaches and stuff. Now, plants, it can go either way. And in particular, the only ones that we know of that are vertically transmitted are grasses. Okay, so grasses do endophytes differently than all other plants. And they're very specific on what endophyte they have. It's this genus called Epicloae. Okay. And it spreads throughout the entire plant, and it makes it into the seed as it's developing. And so grass seed comes prepackaged with Epicloae in it. Every grass you've ever seen. Okay. And you can see it's a well-studied system because grasses include things like corn, wheat, barley, oats, right? So pretty important stuff. If you look at, let's see, this is no endophytes in the top row here, and this is with endophytes in the bottom. So they take these seeds and they basically apply fungicides to make them sterile, like get rid of the Epicloae, and then grow the grass without and with the Epicloae, and then subjected it to stressors like heat, drought, and heat plus drought. What pattern do you see here between A and B?
grass seems to do better under undesirable conditions with the endophytes.
That endophyte is doing something very good for that grass. Absolutely. It's conferring heat resistance, drought tolerance, and even a combination of heat and drought. The grass is doing better when it has that endophyte. Okay.
Wouldn't the endophyte want the host that it's in to die, though, so that it could decompose it? Isn't that the end game?
Well, it really wants that host to make seeds so it can get transmitted to the next host. Does that make sense?
Yeah, that makes sense.
Since it's vertically transmitted, the grass surviving and reproducing is good for this fungus because that'll reproduce it as well.
So if they did the same experiment with the different plant species, would you see the same results?
Probably. Okay, there's got to be a reason that the plants are tolerating having this fungus around. If it wasn't conferring any benefit, it is using a few resources from the plant, or maybe it's, I don't know, it's just not its own cells. So if it wasn't helpful, you would think natural selection would kind of chip away at it over time. What we find, though, is that in nature, it's damn near impossible to find a plant that doesn't have endophytes in it. And so doing natural studies of with endophyte and without endophyte, it's pretty tricky because it's hard to find plants that don't have them. We basically have to do it in a lab. Okay. So this is epicloe in grass. Some of the things that epicloe are making are alkaloids, right? These complex compounds. They're making all kinds of secondary compounds that are protecting the grass from sunburn. So like they're blocking UV radiation. They're making the grass more resistant to pests. These alkaloids are actually like insecticides. Okay. You remember the ergot we talked about that makes your limbs fall off if you eat infected rye? That is right here, claviceps, closely related to epicloe and making a lot of the same compounds. Okay, so epicloe is descended from plant parasites. That's its closest relatives, are these parasitic things. But it's found a way to live in harmony with the plant, essentially using its weird compounds that it makes for the benefit of the plant, and it's getting passed along. And so the better the plant does, the better it does. Okay, so epicloe is a nice monophyletic group that's co-evolved with grasses.
I got a question real quick. Yeah. So obviously animals will eat these plants, right? And they ingest these this fungus as well. Can they also be symbiotic with the host of an animal, or is that just kind of just kind of stay within the digestion tract and just kind of poop it out when they're ready?
Yeah, they're just pooping it out. They're not, to the best of my knowledge, epicloe being really well studied, we have not seen that in this group.
Okay.
They're very specialized for this in which they're found. And because they're vertically transmitted, like each species of grass tends to have its own species of epicloe because it's just been stuck in that grass this whole time. They're not getting out into the animals or anything.
Okay.
Yeah.
Thanks.
So that's vertical transmission. Now, all the broadleaf stuff are dicots. They're not vertically transmitted. They're more like people with our microbiomes. They're picked up from the environment. Okay. This is a recent, just out in July, paper I worked on where we basically went across all the islands of Hawaii and just took the native plants, so things that hadn't been brought over recently, and looked at a bunch of environmental factors and host identity. And the idea was, where are plants getting their endophytes? Like, what determines which endophytes wind up in which plant? And so there was a question from one of you, I forget who, asking, is this host specific? Well, in grasses, yes. Now, in dicots, maybe. There's a strong signal here. You can see the most important explainer for the community was host phylogeny. But there were all these other things that explained it as well. Evapotranspiration, how much sunshine you get, the date, right? Slope, rainfall, elevation. So all these things kind of went in to explain the endophytic community in these plants. So some of it is who you are. So your genotype is going to determine what fungi can inhabit you to some extent, but also where you are. And so in essence, it's kind of the fungi that were there in the soil and leaf litter as you were growing are the ones that you can pick up and tend to pick up. Now, rainfall might also be important for this, right? So we're getting fungal spores dropping in with the rain, that kind of thing. Right, so those are the two ways that plants pick up endophytes from the environment. But once they get there, no matter how they got there, they're doing some similar sorts of things. They're altering aspects of the plant's phenotype. Okay. In this case, the endophytes are altering heat and drought resistance, right? They're improving it. In other cases, they're altering things as fundamental as flowering time. So here's a study I did, essentially just looking at reproductive success in Arabidopsis, kind of the plant model organism. And we did that by measuring how long it took them to go from germination to flowering and how many seeds they put out. We did sterile controls and then these 4 different endophytes in different combinations. And this is days to flower. And look at the stark contrast. So what's going on? What's the pattern and maybe what could explain it? No thoughts? What are endophytes doing to these flowers?
Are they giving off some type of chemical that's like either helping them not get eaten by herbivores or maybe protecting them from insects or something like that?
Yeah, maybe. These are all grown in like a sterile growth chamber with no insects around. But this is the distribution of days to flower. It was roughly 30, 32 days it took to go from seed to flower in this plant without endophytes. If you add endophytes, it tacks on an additional 5 or 6 days. So the endophytes are delaying the time to reproduction in these plants.
So, Dr. Zong, is the reason that that's happening is because, like, I know we're talking about like the symbiosis and stuff, so it's like actually helping it. So is it because it's helping it delay its flowering process so that when it actually flowers, it's much more ready to be flowered, or it's more mature so it can fight off stuff rather than flowering too early?
Yeah. And we saw the same sort of pattern. We did this under normal and drought conditions. And we saw that the ones that if a plant is stressed, it's going to try to flower as soon as possible, right? Let's just get this over with, make some babies so I can hurry and die, right? And so if you subject plants to drought, they tend to flower faster. These helped prevent help confer drought resistance and let the plant basically spend more time putting energy into its seeds. When you measured these seeds, the ones with endophytes, the seeds were actually bigger on average than in the sterile control. And so it was good for the plant. The plants did better with these endophytes. Days to flower was just an interesting proxy for reproductive allocation. In other words, how long are they spending turning air into seeds, right? The ones without the endophytes were 3 to 5 days earlier, meaning they spent less time. They were trying to die faster, essentially. Because in this plant, they make seeds and then they die. Okay.
So am I reading this graph right? Where if you look at the sterile control one, where it's kind of is blunt on the left side, and then it just is that just how many how many flowers actually bloomed? Is that what I'm reading?
The width of this here,
yeah,
the height of it, is the number of plants that took that long to flower.
Which makes it interesting, because if you keep on going down the chart, it just seems like if, in contrast to the sterile condition, all the other plants, it started off slower and then it gradually went more. Does that make sense?
Yeah, and each one of these 4 colored ones, that's a different endophyte that we put in there. What I found really interesting was these are very different, like phylogenetically far away from each other, endophytes. And it didn't matter which one was in the plant. It still basically kind of did the same thing. It delayed flowering overall. And so
it just seems like it gradually grows and then it slowly tapers off, where the sterile control is like, bam, it's here, and then bam, I'm done type thing.
Yeah. So that blunt end on the left and right means that no plants flowered before day 28 or after day 33 or 34. Okay. And here, none of the plants flowered before day 34, all the way down. But what I took away from this was flowering time is something that we measure in plants a lot, and we consider it part of the plant phenotype, right? And so when you're describing a plant phenotype, even in a guidebook, you'll say like days to flower, right? You see it in a seed catalog, like, you know, hybrid 100 early tomato, right? 36 days to flower. Like, that might not actually be a plant phenotype. It might actually be the fungus phenotype, similar to the taxus tree, where we thought part of its phenotype was this anti-cancer drug, but it turns out that was the fungal symbiont. Okay, the story is rarely this simple, because endophytes interact with a lot of different stuff, right? They're interacting with environmental conditions, with the plant genotype, with other pathogens that are showing up. Okay, if a pathogen shows up on a plant that has an endophyte in it, that endophyte might fight it off by producing some new chemicals, which might actually stress the plant out, or it might fight it off by telling the plant to start this jasmonic acid pathway, right? It's kind of like warning the plant, hey, there's a pathogen here, and so the plant responds. So it's this really complex mesh of a lot of moving parts moving together. And rarely is it like a simple, obvious answer, but I've got one more simple, obvious story for you here. This is an endangered plant, Phylostegia kaalaensis, and this is basically showing mortality. So the higher these points are, the greater percentage of these plants have died of this invasive disease. Okay. The study we did was basically to try to provide endophytes for these plants, to engineer a microbiome for them that would help them survive out in the wild. And so we made some endophyte cultures and put those on there, and we looked at the control group and just basically I grew these in a little growth chamber and put an infected leaf in the air intake. So they all were going to get sick with this powdery mildew. And the ones that didn't get sick or got less sick over time were the ones that I'd sprayed with this leaf slurry. So I just went out into the woods, found some related healthy plants that were actually surviving, put them in a blender with some sterile water, and then just sprayed that on these plants. And they did better. They were disease resistant. Okay. When I went back in to find out why, it was a nice clear signal looking at pathogen load, so what the percentage of leaf that was infected versus this particular fungus, Pseudozyma aphidus, which is a little yeast that basically lives on the surface and kind of grows inside the plant. That was the one species that was preventing infection. I didn't know that going in, and so when I was growing endophytes to put on these leaves, I didn't think, let's look for this weird Pseudozyma yeast and spray that on there. But it happened to be in the slurry. And so this is just showing every now and then we get a clear signal for what an endophyte is doing. In this case, this particular endophytic yeast was attacking and killing the powdery mildew as it landed. So a powdery mildew spore would land on the leaf, that would kick on some unknown pathway in this yeast, and it would move from a yeast to a dimorphic hyphal growth form, surround the pathogen cell, and just eat it. And so plant microbiome research is a big field right now, multi-billion-dollar industry. Okay. We can almost say that the definition of an endophyte shouldn't be latent decomposer or non-pathogenic thing living in leaf. I think maybe a useful definition would be a fungus that modifies plant disease. Okay. And you touched on this briefly, like the order of arrival can even matter. Plants or endophytes do modify plant diseases, but it kind of depends on when they got there. And so this is looking at order of arrival. This is just our pathogen, P1, shows up on day 1, okay? And here is the pathogen load. Now here's the pathogen showing up on day 1 and the endophyte showing up on day 5. The disease is worse. If the pathogen and the endophyte show up together on day 1, disease is better. If the endophyte gets 5-day head start, disease almost doesn't happen when the pathogen shows up. So what could explain this potentially? Pathogen's there, 5 days later an endophyte shows up, the disease is horrible. But if the endophyte's there, 5 days later a pathogen shows up, disease doesn't happen. Any guesses? I don't have an answer, so any guesses are appreciated. Maybe we could test this once labs open up.
Maybe it's the endophyte is like, if it's already there and then something new comes in, it's like, no, I need to protect the plant from this. But if the pathogen was already there, the endophyte could, anthropomorphizing it, think that that pathogen was part of the plant and his friend, basically.
Oh, Yeah, it could be that. It could be that like the pathogen sees the endophyte show up and thinks, "Oh crap, I got to fight for my space," and its way of fighting for space is to kill more of the plant and take. I don't know. I think it'd be interesting to test. I have not seen it tested. Well, we've noticed the pattern, right? The order of arrival really matters, but I have not seen a tested hypothesis explaining how this happens. So there's a lot going on out there. Let's skip this next slide.
I mean, it also could be that the disease was just more well established if it was there before the endophyte got there.
Yeah, but here it showed up like on day 1, and then the endophyte got there on day 3. Okay, so you're saying if it's more established, but what freaks me out is that the disease is actually worse after the endophyte shows up than if no endophyte showed up at all.
Yeah, that's weird.
I think it's super weird.
Dr. Zahn, couldn't
Yeah.
it just basically be like the disease gets there, they don't see anything, so they're just like, just doing their thing. But then once it sees there's anything else to do, it just goes full speed on us, I guess.
Yeah,
Or I guess on the plant.
it might be like a competition thing, like
Yeah.
Just like if the endophyte's there and it sees a pathogen, it's like, no, right? So maybe the pathogen's doing the same thing, but its way of competing for space winds up killing the plant more since it's a pathogen. That could be the case. I'd be interested in testing that. Okay, so we'll finish up with this slide here. I just want to point out what a big field of study endophyte research is for conservation and especially for agriculture. This company here, Indigo Ag, okay, it was a startup. It's maybe like 6 or 7 years old, and it's already, topping the charts at $10 billion valuation. And their entire thing is engineering endophytes for agriculture. And so they are trying to find magic bullets or custom tailored endophytes for specific regions and specific crops that reduce your need for pesticides, reduce your need for watering, increase drought resistance and heat tolerance and all that kind of stuff. And you can see their plants do better than the ones that aren't treated, and that's just a microbiome treatment right there. So in the non-lucrative world of ecology, the lucrative section is plant-associated fungi, because agriculture is a multi-billion-dollar industry. So if you're into mycology, that's probably the field to go into. Not necessarily human mycology or pathogens, because, well, as you saw, the US isn't even participating in incentivized research plan for human pathogens. So move to Denmark or get into agriculture if you're going to go into mycology. All right. So reading assignment this time is Christian et al., 2017. And this is a really fun paper. Exposure to the leaf litter microbiome of healthy adults protects seedlings from pathogen damage. Okay. So this is important for you if you like chocolate, because cacao trees. And so she worked on, as a grad student, this paper basically trying to figure out what was going on with the endophytes in cacao trees and how it helped them prevent or resist disease. So it's a lot easier of a paper to read than the last one, probably. And it's important if you enjoy hot cocoa. Any questions?
Yeah, so the second exam's due on Monday, right? The 26th?
Monday. Let me triple check here. October 26th, 11:59 p.m.
Great.
Yep. And I will get the quiz for Christian et al. Posted. And I'll go ahead and post up the aquatic eDNA protocol that I use. Real low cost method for sampling from water. Okay. All right. I'll hang out for a couple minutes if people have any questions. Otherwise, get out of here.
Oh yeah, are you going to be in your office at like between 3 and 4 today?
Yes.
Okay, well, thanks.
Hey, Dr. Zong, can I ask a generalized question regarding the test?
Mm-hmm.
So the one question that's regarding you have to like use that one search engine to find stuff.